Okay, good evening everyone. So let me know whether I'm audible to you or not, because invisible is not possible. We have a power cut problem right here. So please let me know through this technical poll whether I'm audible to you or not. Okay, so let's start our today's topic. So in our today's topic, we are going to discuss the previous year questions of biotechnology, principles and processes. In the last 2 days, we have already covered the questions of the principle of inheritance and molecular basis of inheritance, right? So today we are going to cover some previous year questions of this biotechnology, principles and process chapter. So let's start our today's class without wasting any time. So the first question for our today's class is the DNA molecule to which the gene of interest is integrated for cloning is called. You have to give the name of the molecule in which we integrate our DNA so that we can have the gene of interest recombinant to that. Your option A says template, option B carrier, option C transformer, and option D vector. So here I'm launching the poll. Those who are not able to answer the poll, answer in the poll, please give their answer in the chat box. Okay. So what you call to that molecule in which we integrate our gene of interest? A template, B carrier, C transformer, and D vector. Okay. So as everyone has given the answer, so this is your result. Okay, let's find out. As we know, the molecule, or we can say the component in which we incorporate our gene of interest. That is also a DNA molecule of some other organism, right? So that organism which is carrying our gene of interest, or we can say we are integrating our gene of interest for cloning, right? Here we are inserting our gene of interest for the cloning purpose. So cloning is the main. So instead of using the template is the strand of DNA which is getting or replicated, right, or transcribed. Carrier is there. So carrier, that means just carrying the DNA. It is not actually replicating it or making a clone. Transformers, who are able to transform itself, who are able to get transformed. So only answer we have is the vector. Vectors are those DNA molecules which are able to carry our gene of interest for the cloning, right? Carriers are just to travel, just to take your gene of interest from one place to other. But here we have included the carrier. That is why we can say the vector is the correct answer. Okay. So next question here is the cutting of DNA at specific location become possible with the discovery of which molecule? The cutting of DNA at a specific molecule become possible. Your option are like A, selectable marker, B, ligases, C, restriction enzyme, and D, probes, right? So as you have already given your answer in chat, yes. So the answer is C, yes, the restriction enzyme, because these are the only enzymes which are able to recognize the specific sequence, right? They are able to recognize the specific sequence. Can anyone tell me what this specific sequence which is recognizing known as? What are these specific sequence are known as? Very good. They are known as palindromic sequence, or we can say those are the recognition sites. The specific location is a recognition site, right? So answer is C. Why we have not chosen the selectable marker? Because this is a gene which is coding for some specific enzyme in which we incorporate our gene of interest, so that we are able to find out that the incorporation has taken place or not. Ligases are the enzymes which are used to ligate. That means to stick to stuff together, or can say to make a bond. Whereas probes are used to magnify something, to visualize something. Okay. Next question says, you have to find out of these four statements, or out of the four options, which one is incorrect. Some matching, the wrong matching among these four options. So here I'm launching the poll. You can give your answer through this poll. Option A says somatic hybridization, which is the fusion of two diverse cells. Option B says vector DNA, site for the tRNA synthesis. Option C says micro propagation, in vitro production of plants in large number. And option D says callus, that is unorganized mass of the cell produced in tissue culture. Okay, so as you have given your answer, so this is your result. Okay, let's find out which statement is correct. Okay, so option A says that is somatic hybridization. Somatic, those cells who are not forming the gametes. Hybridization, that is combination of the two different stuff. Right. So itself, its statement is saying it is a fusion of two diverse cells. So we can say yes, the option A is correct. Option C says micropropagation, which is also known as tissue. We are growing them in a culture medium, right? A specific culture medium. So, and in the presence of a specific culture medium, those meristematic cells start growing, and they form some internet issue. This meeting is leaving again. I'm so sorry. All the three options are correct. Only left option is B, which is saying vector DNA is site of tRNA synthesis. No, vector DNA is DNA in which we incorporate our gene of interest. So yes, only option B is the incorrect one, right? So very clearly, you are performing so well and you have given a correct answer. Okay, so let's move to the next question. So next question says, which one of the following technique made it possible to genetically engineer living organism? Which technique made us possible to genetically engineer the living organism? Option A, recombinant DNA technology. Option B, X-ray diffraction. Option C, heavier isotope labeling. And option D says the hybridization. So now you have to find out which is the correct answer. Okay, very good. So you have given your answer in chat box. That is right. That is recombinant DNA technology. Only by using the recombinant DNA technology, we are able to genetically engineer living organism. See, the recombinant DNA technology itself, or we can say biotechnology itself, contains the three. It contains the three fields, right? The genetic engineering, gene transfer, and gene cloning, right? So all these three are the parts of recombinant DNA technology, which is used to generate organics. We are doing the modification with the DNA molecule. So yes, option A is the correct answer. Question number 5, your next question. Which of the following are used in gene cloning? Out of these four options, which one molecule is used in a gene cloning? A, nucleoids, B, C, mesosomes, and D, plasmids. So you have the correct answer. E, plasmids. Below these plasmids, they are DNA. So this self-replication is so sorry. It is getting disconnected again and again. Okay, now move to the next question. Here, question number 6, which is manipulation of DNA is in genetic engineering become possible due to the discovery of, by the discovery of which molecule the, we can say, the genetic engineering become possible. So your options are A, restriction endonuclease, B, DNA ligase, C, transcriptase, and D, primase. So what is the correct answer? Yes. As we have seen, it is also given in our NCERT, only after the discovery of restriction endonuclease through the bacterial cell, we are able to perform the genetic engineering. Because these restriction endonuclease, we have three types of restriction enzyme. Out of this restriction endonuclease are the ones who are able to recognize a particular sequence and cut in between the DNA, and not only the one strand of DNA, they are able to cut both the DNA strands, right? So yes, option A is the correct answer. Now we are moving towards our next question. The next question is question number 7. The bacteria generally used for genetic engineering is option A, Agrobacterium, option B, Bacillus, option C, Pseudomonas, and option D, Clostridium. You have to give your answer in chat box or either in a poll which we are launched. You have to find out the name of the bacteria which is used for the genetic engineering, right? Okay, so as you have given your answer, some have given your answer B. Okay, so your answer is A. So let's see. As you know, the agrobacterium are the easily engineering bacterium, right? We are not using bacillus. Bacillus is used for making the or introducing. We are using bacillus species, or we can say genus, just to introduce the toxic gene. The example is Bacillus thuringiensis, right? From which we were making the BT cotton. So the answer is agrobacterium. The correct answer for this question is agrobacterium. Now your next question is, which of the following is related to the genetic engineering? A, heterosis, B, mutation, C, plastids, and D, plasmids. So out of these four terms, which one is related to the genetic engineering? You can give your answer in chat box. Okay. So you have opted for option D. Yes. So it is a correct answer. Option D, plasmid, because without plasmid, we are unable to make a recombinant DNA, and we are also not able to make the clones of our gene of interest, because plasmids are the one who have the replication site, that is ORI, and they have the tendency to self-replicate. Very good. So your next question given here is, the genetic engineering is possible because. How the genetic engineering is become possible? You have to find out the reason. Option A says we can cut DNA at specific sites by endonucleases like DNase 1, right? Option B says the restriction endonucleases purified from bacteria can be used in vitro. Option C says the phenomenon of transduction in bacteria is well understood. And option D says we can see DNA by electron microscope. Okay. So in genetic engineering, basically our motto is to develop the hybrid cells, right? We have to develop or grow the genetically engineered cells in vitro condition, right? And that can only be possible when we have all the enzymes which are able to work in vitro condition, because in vivo we are unable to control the each and every stage of our experiment. That is not become in under control, because at that time when we are doing something in vivo, the body of an organism is also playing the role. So we can say the restriction endonuclease purified from bacteria can be used in vitro. That this enzyme, the most important enzyme, because this is the only enzyme which make our genetic engineering possible. If it is able to work in vitro, then yes, it is the main reason through which the genetic engineering become possible. Very good. Yadagiri, you have marked option A. See, beta, we can cut the DNA at specific site by endonuclease, like DNA is one. It cannot be the correct answer. We can cut the DNA, right? But it is working in vivo condition, right? And in vivo condition, we cannot have any control. So we want something which can be done in vitro, right? Because some of the steps require our manipulations, right? Some of the steps required are control. So that is why we are looking for the enzymes which are able to work in vitro condition. Is it clear to you, beta? Bache? Okay. So let's move to the next question. The next question says, when scientists make an animal superior by viewing of genotype, here we are talking about the genotype, introducing some foreign gene in it is called. Here we are introducing a foreign gene in the genome of a particular animal to make it superior, right? So out of these four, which technique we are using? A, immunization. B, tissue culture. Sorry, A, immunization. B, genetic engineering. C, tissue culture. And D, biotechnology. So you have to give your answer in this poll, that out of these four techniques, which is used to make animal superior by incorporating the foreign gene in it. Okay. So here I am ending the poll. Okay. So see, here we are just talking about the incorporation of, right? We are not talking about transfer of gene either, or cloning of gene, right, in a sterile condition, or the production of the product from that gene of interest. We are just talking about the introduction of foreign gene in the genome of our animal of interest, right? So out of these four, immunization is the activation of your immune system. Tissue culture, in that we are just growing a same kind of plant by, or we are just growing the number of cells by providing a specific culture medium. Here we are not doing any kind of mutation or manipulation. In biotechnology, we always talk about the production of the product, right? Apart from introducing the foreign gene, we have several steps involved. So only genetic engineering is the technique in which we are doing something engineering. Yani, that means we are making something, bonding, we are breaking something, we are rejoining something. So genetic engineering, yes, is the correct answer, and it is the technique which is used to introduce a foreign gene into the genome of our animal of interest. Okay. Now, moving to the next question. The next question says, which of the following organelle is related with the genetic engineering? Now we are talking about the organelle. So out of this, which is related with the genetic engineering? Option A, mitochondria. Option B, plasmids. Option C says Golgi bodies, and option D says lysosomes. Yes, as you know, we always perform the genetic engineering in the simplest organism. Usually we are taking bacteria. In those bacteria, no specific organelle is present except the extra chromosomal plasmid, that is double-stranded circular DNA. So we can say the answer will be option B, because they are almost prokaryotic organism. So mitochondria, Golgi bodies, and lysosome cannot be possible. Next question says, identify the wrong statement. You have to find out the wrong statement, the statement which is incorrect. Please keep this thing in mind while reading the options. You have to find out the wrong statement, right? Don't go for the correct one. With regards to the restriction enzyme, now you have to find out the incorrect statement regarding the restriction enzyme only. Okay, so your option A says each restriction enzyme function is inspecting the length of a DNA sequence. Option B, they cut the strand of DNA at a palindromic site. Option C says they are useful in. Option D says the ends can be joined by using.
Pura DNA ಬೇಕಾಗಿಲ್ಲ ನಮಗೆ ಅದರ sequence length of DNA sequence.
AB Yadagiri, please mute your mic first. Okay, now you have to find the correct option, the incorrect one, right? Okay, so you have opted for option A. Let's find out. Start with the D then. The option D says sticky ends can be joined by using DNA ligase. Okay. Option C says they are useful in genetic engineering. Right? Both the statements are correct. Option B says they cut the strand of DNA at palindromic sites. Right. But option A says each restriction enzyme function by inspecting the length of DNA sequence. No, they are not inspecting the length of DNA sequence. They are just inspecting a particular sequence of the DNA, right? So only option A is giving a incorrect statement. So yes, option A is the correct answer for our question. Okay, let's move to the next question. The next question is choose the correct pair from the following. You have to find out the correct pair now. Now, look very carefully, read it very carefully. You have to go for only the correct one. Option A says ligases. They are used to join the two DNA molecule. Option B is polymerase, which is used for breaking down the DNA into the fragments. Option C says nucleases, which separate the two strands of DNA. And option D says exonucleases, which make cut at specific position within a DNA. Okay, so we have to find out the correct one. So let's find out. As you know, exonucleases cut the DNA not at a specific position. They are cutting it at the ends. So it is not a correct statement. This is incorrect. Nucleases are used to digest the complete nuclear material because the itself is nuclease, something which is breaking down, as ase is later written, something which is breaking down the nuclear material. So nuclear material is DNA. So it is not separating, it is just breaking it down. So it is also not correct. Option B says the polymerase are the enzymes which break the DNA into fragments. No, they are the enzymes which are used to synthesize the fragment of DNA. So it is also incorrect. So yes, only one option, that is ligases, which is used for the joining of molecule, is the correct answer. So yes, option A is correct. Now your next question. In this question, you have to find out the specific palindromic sequence. Out the sequence which is recognized by the EcoR. So we know the EcoR1, the sequence which is recognized by the EcoR1 is GAATTC. Okay, GAATTC, because from the both sides it has to be read same. That is why it becomes palindromic. So we have to find out this sequence only, and out of these four options, so let's start with option A. So here it is written GAATTC. Yes, this is what we are looking. Now move to backward. Now study the second stand backward. GAATTC. Yes, so both the strands are making the palindromic sequence, and yes, this is the sequence which is recognized by the EcoR1. So option A is the correct answer. Very good, Yadagiri, you are doing very well. Okay, and those who are not able to give their answer through poll, they can give their answer in chat box. The sequence that control the copy number of the linked DNA in the vector is termed sequence. We are talking about the sequence of the genome which controls the number of copies, the number of replicated copies formed inside the vector right? We have to control the number of copies of replication. So here we have the 4 different sequences which are usually found in vector. So out of these 4, you have to find out one. A, selectable marker. B, ORI site. C, palindromic sequence. And D, recognition site. So you have to find out of these 4, which one is responsible for controlling the replication. Yes. So the replication, the ORI site is known as origin of replication, right? Origin of replication. It is the only site which is working as a switch for controlling the replication. So option B is the correct answer. Okay. Next question we have, in gel electrophoresis, separated DNA fragments can be visualized with the help of. How can we visualize the DNA fragments from the gel electrophoresis? Option A says acetocarmine in blue, bright blue light. B says by using the ethidium bromide in UV radiation. C says acetocarmine in UV radiation. And option D says ethidium bromide in infrared radiations. Right? Okay. So as you have given your answer, we know the visualization. For visualizing the DNA, we put the ethidium bromide because we have to find out the segregated DNA fragments. We initially put the ethidium bromide in the DNA gel while making it. So whenever the DNA gets segregated, the ethidium bromide binds it and makes a complex. When it gets exposed by the UV radiation, only then they make the bright, which color bands they form? When we expose the ethidium bromide under UV radiation, which color bands they form? Can anyone tell me which color bands are formed? Green, red, orange, blue, any of the color which you like to tell? Okay, so whenever the ethidium bromide binds with DNA and the UV exposure takes place, it shows the orange color bands. It shows the orange color bands. Okay. Now move to the next question. Here is the next question. It says, following statements describe the characteristic of the enzyme restriction endonuclease. So here we have given some characteristics of restriction endonuclease. Identify the incorrect statement. We have to find out the incorrect statement, the statement which is not right, right? So option A says the enzyme recognizes a specific palindromic nucleotide sequence in the DNA. Option B says the enzyme cuts DNA molecule at identified position within the DNA. Option C says the enzyme binds DNA at specific sites and cuts only one of the 2 strands. Option D says the enzyme cuts the sugar phosphate backbone at the specific site on each strand. Now you have to choose your option. You have to find out the incorrect statement. Please keep this thing in mind. You have to find out incorrect statement regarding the restriction endonuclease. You know, restriction endonuclease is an enzyme which is responsible for cutting the DNA at both the strands, and they always result in the formation of either sticky ends or the blunt ends, right? So give me your correct answer in the chat box, or you can give your answer in this poll. So I'm launching the poll. Here is your poll. Please give your answer. Okay, so you have opted for option C. Let's read this option for once again. The enzyme binds DNA at specific sites and cuts only one of the two strands. Yes, this is incorrect statement, because when we see this is the enzyme, this is our DNA fragment, and when we apply the restriction line, it will result either into the sticky ends, right, or into a, okay. Whenever the sticky ends or blunt ends are forming, they are cutting both the DNA strands, right? So here it is written only one strand. So it is a incorrect statement. So the incorrect statement we are looking for. So option C is the only answer. Next. Okay, so question number 18 is here, and it says a selectable marker is used to help in eliminating the non-transformants so that the transformants can be rewritten. Here we have to find out the statement regarding the selectable marker now. So option A is help in eliminating the non-transformants so that the transformant can be regenerated. Option B says identify the gene for a desired trait in an alien organism. Option C says select a suitable vector for the transformation in a specific crop. And option D says make or mark a gene on a chromosome for isolation using restriction enzyme. Okay, you have to find out the correct statement regarding the selectable marker. Please give your answer in chat box, because so sorry, you have to give your answer in chat box, because due to network issue, the poll is not launching. Okay. Okay. So you have already given your answer. You have opted for option B. Let's read it. It identifies the gene for a alien organism. No, the selectable marker is just used. The selectable marker is used as the gene sequence where we can incorporate our target gene so that we can find out by eliminating the production of a particular enzyme. So yes, option B is the correct answer. Okay, just wait a minute, Yadagiri. I have to make some changes. Just a minute. I hope now signal become better. Okay, let's move to our next question without wasting time. So the next question says, given below are four statements pertaining to separation of DNA fragments using gel electrophoresis. Here we are going to discuss the technique of gel electrophoresis. You have to identify the incorrect statements regarding the gel electrophoresis. Now you have to find out the incorrect statements regarding the gel electrophoresis. So here four statements are given. Statement A says the DNA is negatively charged molecule, and so it is loaded on gel towards the anode terminal, right? Option B says DNA fragments travel along the surface of the gel whose concentration does not affect movement of the DNA. Option C says smaller the size of the DNA fragment, larger is the distances traveled through it. Option 4 says pure DNA can be visualized directly by exposing the UV radiation. So we all know the process of gel electrophoresis. Now you have to find out the given four statements. Out of these given four statements, how many statements are incorrect, right? So you can easily do that. So let's find out it fast. Yes. Okay. So let's read it one again. Okay. To see whether your answer is correct or not. The option A says the DNA is negatively charged molecule and so it is loaded on the gel towards anode. No, the anode is itself anode is itself a positively charged. If we load the DNA, which is already negatively charged, at the positively charged electrode, the DNA will not move further in the gel. It will stay only inside the gel and it will move out in the buffer solution. So this statement is incorrect. We got the first incorrect statement. Next says the DNA fragments travel along the surface of the gel whose concentration does not affect. No, the gel concentration always affect as the concentration increases. The pore size decreases. So concentration is inversely proportional to the pore size. So it depends only on our concentration of gel that what length of fragment of DNA can move along it. Okay. So option B is also incorrect here, or statement second is also incorrect. Statement 3 says smaller the size of DNA fragment, larger is the distance it will travel. Yes, because we are continuously decreasing the size of pores in the gel bed. If our DNA is of very smaller size, if the size of the DNA is very smaller, then it will not get stuck within the pores and it will reaches to the end. Okay, so when a DNA is reaching to the end of the gel, right, we can say it is the smallest DNA because it has traveled the longest distance. So yes, option 3, or we can say the statement 3 is correct. Let's find out the fourth one also. Okay, we can do the option checking also from here. Now, as we know, the option 3 is correct. The statement 3 is correct. Now we look at these options. Option A, B and C, all the three are containing statement 3, but statement 3 is correct. So these three options cannot be there. So yes, we can go with the option D without any hesitation. Okay, but it is not working every time. We also have to read the fourth statement as well. So fourth statement was saying pure DNA can be visualized directly by exposing UV radiation. No, we have to use a probe, that is ethidium bromide. The dye was ethidium bromide for the visualization of DNA. Okay, so never go without reading your all the statements. Always read the statements and then mark your option. Okay, this is I have done because the three options are containing the correct statement. So there is no use of reading the fourth one, right? So now your next question, which of the following is commonly used as a vector for introducing a DNA fragment in human lymphocytes? Oh, such a quick answer. Very good. As we are talking about the human, we know we are using two types of vectors. The Agrobacterium tumefaciens for the plants, and for humans we are using retrovirus. So here we are only talking about the humans. So we are using the retrovirus who are non-pathogenic in nature because these retrovirus are carcinogenic in nature. So we make them first the non-pathogenic, then only we introduce them into use them for the introduction of the DNA in a human lymphocyte, or we can say any of the human cell. So now your next question is the DNA fragments separated on an agarose gel can be visualized after staining with. We have discussed this so many times, so I don't think we have to go for reading each and every option. Just look for the answer. And yes, you have given the correct option. You have just chosen the correct option, that is C, because we know the DNA fragment which is separated during gel electrophoresis can only be visualized by the ethidium bromide, right? So we have to just look for the ethidium bromide. There it is given. Okay, so that is our correct answer. Very good, Yadagiri. Okay, so next question says the DNA fragments are. Now find out which statement is correct. What type of DNA fragments are? They are negatively charged, neutral, either negatively or positively charged, depending on their size, or positively charged. Everyone knows that DNA is a negatively charged molecule because it contains the phosphate group and the free hydroxyl group, and both of them have the negative charge. So whatever is the length of the DNA segment is there, whatever length of the DNA segment is there, the charge always remain the negative. So yes, DNA fragment is negatively charged. Okay, the next question for you is a gene whose expression help in identify the transformed cell is known as, we have discussed this in throughout the session several times, that the particular sequence of gene which we use for the identification of transformant is a selectable marker, because this is the section, or we can say this is the portion of the gene which is coding for a particular enzyme. If we make the recombinant, then the particular enzyme will not get coded. That particular enzyme will not make. Instead, only our product of interest will be forming there, right? So you have given the correct answer. That is option D. The last second question for today, because we are going to do only 25 questions. So the last second question says, what is the criteria for DNA fragments movement on agarose gel during the gel electrophoresis? What criteria we are using during the gel electrophoresis of the DNA fragments? Option A, the smaller the fragment size, the farther it moves. Option B, the positively charged fragments move to the farther end. Option C, negatively charged fragment do not move. And the option D, the larger side fragment moves the farther. That is, they travel the longest distance. So we have just discussed the as the small तारीफ कर दी. Okay, so let's complete our session. Okay, something happens again. इस तारीफ पसंद नहीं आई, ये बार बार भाग रहा है. Okay, let's quickly complete this question so that it won't get away again. So a question says a foreign DNA and plasmid cut by the same restriction endonuclease to join to form a recombinant plasmid. Which enzyme we are using out of these given four options to join the fragments of the DNA, the foreign DNA and the plasma? You have option A, EcoR1, B, Taq polymerase, C, I hope you have read the question. Please give your answer. JNV Yadagiri, please give your answer as fast as possible. That which enzyme out of these given four which we are using to join the two different fragments of DNA? Option A, EcoR1, option B, Taq polymerase, option C, polymerase 3, and option D, ligase.